Our immune system is like a quirky cast of characters, each with a defined and unique role, and the sole goal of protecting us and keeping us healthy. We all have our very personal lineup of helpers. But some diseases' specialty is to elude detection, and at times something slips through the cracks. In this episode, we'll delve into medicine's quest to harness the power of our immune system and into the breakthroughs that have made a real difference in cancer treatment. I'm Silvia Lazzaris, and this is The Science of You, a podcast series about the groundbreaking science that's changing the way we approach healthcare. Together with those who are leading the charge, we will journey from the discoveries that transformed our understanding of the human body to the innovations that promise to revolutionize treatments and medical practices. The Science of You is a Cora Media podcast, supported by the European Partnership for Personalised Medicine, coordinated by Fondazione Toscana Life Sciences, and co-funded by the European Union's Horizon Europe Research and Innovation Programme.
So, immune system is a complex system that is formed at the same time by cells and at the other hand by different substances that are present in our blood and also on the surface of our skin and mucosa. Cells that are part of the immune system, they recognize microorganisms that can induce infections and can ingest and eliminate them. At the same time, they produce different substances such as antibodies or defensins that are acting directly against microorganisms like a sort of natural antibiotics.
This is Professor Mihai Netea, an infectious diseases specialist at the Radboud University Medical Center in the Netherlands. He's here to infuse some solid science into the metaphors I'll bring about to make sense of how our immune system works. Here we go. Imagine your body as a bustling neighborhood, with cells going about their daily business, delivering nutrients, managing repairs, and generally keeping the place in order. Like in any thriving community, there are occasional troublemakers, some nasty germs, for example, that threaten to upset the peace. Luckily, some cells take on the important role of vigilant neighborhood watch. Dendritic cells are the first to notice anything out of the ordinary. They spring into action quickly and peacefully. Their job is to send out signals, alerting other cells in the area to potential trouble. T cells, the neighborhood patrollers, respond to the dendritic cells' alarms. They don't rush in blindly. They're discerning and thorough. They check the ID badges of other cells, ensuring everyone belongs and is doing their part. But if T cells discover something that seems off, they promptly alert the B cells, the neighborhood's planners, which step in to prepare defenses by producing antibodies that stay on high alert for any unwelcome guests. Each type of antibody is like a specialized security detail, finely tuned to recognize and even to physically bind to a specific intruder, ensuring that it can neutralize the threat. Essentially, eliminate it. Once the antibodies have done their vital job, they call in the cleanup crew, the macrophages. Their task is to clean away any debris, dead cells, rogue bacteria, or anything else that doesn't belong to this well-maintained community. It's a remarkable operation, and each of us has their very own coordinated protection squad, tirelessly working to keep these bodies of ours safe and sound.
The immune system has a number of very important functions. One and most obvious one and most studied probably is that of eliminating infections. But in addition to that, there is also a very important role of immune cells for immune surveillance against cancer cells that develop continuously in our body, and also for tissue repair.
Because sometimes trouble isn't a loud invasion or a wild street fight. It's more like a sneaky figure slipping into the community. Someone who blends in like family, trusted by everyone, so no one suspects a thing. But their true aim is to cause trouble. And often, by the time the neighborhood watch realizes they need to step in, the damage is already done. In our metaphorical city, these stealthy intruders go by one name: cancer.
In terms of cancer cells, they occur unfortunately very often in our body, and they are also recognized and eliminated by immune cells in a process which is called immune surveillance. Indeed, what is also very important to mention is that in the end, cancer cells are cells of our own body. So it is much more difficult for immune cell to discriminate between a cancer cell and a normal cell of our body, whereas microorganisms are very alien, are very different. And because of some of the cancer cells look very much alike our own cells in the body, they are allowed actually to escape this process of immunosurveillance and developed malignant processes, cancers basically, that sometimes unfortunately become very severe and can kill a patient.
That's the problem with cancer. It manages to slip past our immune system. Back in the late 1800s, Dr. William Coley, an American bone surgeon and cancer researcher, made a curious observation. He noticed that some patients suffering from infections experienced what could only be described as a miraculous vanishing act, meaning that sometimes, if they found themselves fighting both cancer and an infection, their cancer would simply disappear. Coley thought that this could be because the infections would suddenly awake the immune system, and dedicated his career to supercharging the body's defenses against cancer. But Coley was a man ahead of his time. His treatments would have required individual tailoring, or at least much more scientific rigor, which would have been impossible with the tools available at the time. Meanwhile, the dawn of the 1900s brought the glimmering allure of radiotherapy, which captured everyone's attention far more than Coley's eccentric immune-boosting theories. As a result, his work faded into the background for almost a century. Michele Maio, a full professor of medical oncology and director of the Center of Immuno-Oncology at the University Hospital of Siena, explained to us how the revival of this idea unfolded.
It was understood that the cells from the immune system talk to each other through a number of cell surface molecules, which are crucial for the activation and the deactivation of an immune response. So based on this knowledge, the idea has been, if we can tackle these mechanisms of the immune system, can we unleash the activity of the immune system that can be blocked in some cases? And this unleashing of the immune system allows us to take control also of cancer cells. So this was basically the idea. And this idea has been developed on the studies of two investigators that are Jim Ellison and Tasuku Honjo, who first identified two cell surface molecules that belong to the growing category of the immune checkpoints, demonstrating that these molecules, CTLA4 and the PD-1, PD-L1 axis, are crucial in shutting off the activity of the immune system.
Let's break it down in simpler terms. James Allison, an American immunologist who still runs a cancer research lab in Berkeley, and Tasuku Honjo, a professor of immunology at the University of Kyoto, became independently intrigued by T cells way back in the 60s and 70s, and independently decided to dedicate their lives to figuring them out. From our neighborhood watch metaphor, T cells are those patrollers that check other cells' ID badges. Back then, the scientific community had a vague understanding that T cells could roam through the body trying to find rogue cells, and then calling for help from the rest of our immune system. Beyond that, we knew nothing. And both these men were eager to understand more. Through their research, they uncovered a fundamental mechanism that explains how cancer sometimes slips past our immune defenses. Here's how it works. Once they've found some intruder, T cells decide how many antibodies the B cells, the manufacturers, should make to fight it off. But sometimes they overestimate how many they need. When this happens, the body ends up with way too many antibodies. Enough to start attacking everything, even the healthy cells that are just minding their own business. It's like calling in a huge crowd to handle one intruder. And suddenly, everything is in chaos. This chaos is called cytokine release syndrome, where the immune response becomes overly aggressive and can cause significant harm to the body, even death. To prevent this, our immune system has its own built-in safety net. Allison and Honjo discovered what makes T cells hit the brakes. They call them checkpoint proteins. These proteins play a crucial role in keeping our bodies intact against big immune overreactions. But they can also throw a wrench in the works when it comes to cancer. If they stop T cell responses too early, before every last tumor cell is eliminated, the cancer will persist and eventually win over. The idea that struck Allison and Honjo is, can we find a way to block those checkpoint proteins, allowing T cells the time they need to finish off all the tumor cells once and for all? That's when immunotherapy was born. A type of cancer treatment that enhances the body's immune system to recognize and attack cancer cells more effectively. Immunotherapy boosts or modulates immune responses, making our immune system more effective at identifying and destroying cancer. The first clinical trial took place between November 2000 and October 2002, including patients with metastatic melanoma.
Melanoma, cutaneous melanoma, actually, is a cancer of the skin that arises from the skin. And until 10 years ago, we had nothing to offer to patients with advanced melanoma that could indeed impact on their overall survival. So basically, the 5-year survival rate of patients with melanoma was roughly anywhere between 5 to 10%. 5-year survival. We now know that the 10-year survival of patients with metastatic melanoma treated with immunotherapy is over 50%. So this is the most impressive piece of information that has been generated. And starting from melanoma, then immunotherapy with checkpoints in this case has moved both to the earlier phases of the disease, but also to practically any tumor type.
The discovery of immune checkpoint inhibitors has sparked a line of research with the potential to cure various types of cancer. This groundbreaking work earned Allison and Honjo the prestigious Nobel Prize in 2018, which recognized their significant contributions to the field and the hope they've brought to countless patients.
After melanoma, the research has moved to, for example, lung cancer. Now we know that at 3 years and 5 years, we have roughly 30% of patients with lung cancer that survive their disease, patients with metastatic disease, and then so on for many other tumor types. Of course, there is much work to do, there is much to learn, there is much to achieve. But, just by thinking of these numbers that I just mentioned, the future means that we can certainly obtain much better results.
And several other promising immunotherapy treatments have been developed at the same time.
CAR-T immunotherapy is also very important. It makes use of engineered T cells that have special receptors built in on its surface, which make them much more easier to recognize cancer cells.
These are therapies designed to help T cells, again, our patrolling cells that go around to check if everything is running smoothly, to better identify cancer cells. Back in 1992, Michael Sadelain, a researcher at the Massachusetts Institute of Technology, began dabbling with the latest genetic engineering tools, dreaming of crafting some supercharged cancer-fighting T cells. Fast forward a couple of decades, and he teamed up with Professor Carl June from the University of Pennsylvania to develop a therapy known as chimeric antigen receptor, also called CAR-T cell therapy. In the simplest possible terms, here's how it works. First, doctors take a sample of your blood and collect millions of T cells. Then they genetically tweak these cells, giving them a shiny new receptor that can latch onto a specific protein found on cancer cells. Once that's done, they reinfuse the modified T cells back into the body. When these cells encounter their designated cancer protein, they signal the rest of the immune system to unleash hell on those cells. But as it often goes, there are a couple of hiccups. The first one lays in identifying the right protein target. The issue is, some proteins that would make ideal targets are also present in vital tissues. This means that while the T cells are trying to take out the cancer, they might inadvertently cause collateral damage to healthy cells. We're back to that cytokine release syndrome we were talking about earlier. The pharmaceutical sector is working to navigate this tricky terrain by honing in on a unique protein called Claudin-6. This protein is found in fetal tissue and in certain cancers, but thankfully not in healthy adult tissues, making it a promising target. As an alternative, researchers are exploring ways to make T cells a bit smarter by engineering T cells with multiple receptors. A bit of a logic map that makes the cells spring into action only when specific conditions are present. The other limitation of CAR-T cell therapy and immunotherapy more in general is that these treatments don't work for all types of tumors. And not every patient is a good candidate. So immunotherapy is not a magic solution for everyone.
So immunotherapy has been successful in a number of solid cancer, such as melanoma, such as also lung carcinoma, but not in other type of cancers, and we don't know exactly why.
The challenge isn't just that these therapies work better for some cancers than others. They also work better for some people than others. And once again, we come back to the recurring theme of these episodes, the uniqueness of our bodies. So, while researchers are striving to crack the code of cancer treatment across different cancer types, they're also navigating the complex terrain of individual biology, working to make these therapies effective for everyone, not just a lucky few.
Each patient is different, each individual is different, and it reacts differently to the same problem. In such a way, for example, we have blonde hair or brown hair and so on, and this is what we see on the surface, but in the same way, our immune cells are different and act differently when they are encountering a pathogen in an infection or a cancer. And we have to measure that and approach specifically in a personalized manner.
The thing is, each person seems to have their unique immune system. Albert Heck, a professor of chemistry and pharmaceutical sciences at Utrecht University in the Netherlands, together with his colleagues, stumbled upon this immune diversity while studying antibodies in the blood of both healthy and sick individuals. Their findings could shed light on why some people react differently to COVID vaccines. Before 2021, scientists had estimated that the body churns out billions of antibodies at any given moment, a number so vast that it would be immeasurable. But this research team decided to take a closer look using mass spectrometry, a technique that separates substances based on their molecular makeup. In other words, they could measure antibodies individually, and the results were surprising. First, they found out that in the blood of both healthy and sick people, only a few tens of thousands of distinct antibodies were present in high amounts. Second, they discovered that our immune system responds to germs in wildly different ways. And each person's antibody profile is unique, like a fingerprint. So if we go back once again to our neighborhood watch metaphor, think of it this way. Each of us has our own crew. They all have their unique faces and personalities. This individuality explains why we have our own Achilles heels, often getting reinfected with the same viruses or bacteria, no matter how many times we've battled them. Mine, for example, is the Streptococcus infection. Hello there. That happens because our antibodies responsible for fighting off those Achilles heel invaders may be less effective than the antibodies responsible for fighting off other stuff. And that's why a personalized approach is likely the way to go.
So in each patient, we have to measure, assess the immune system, and apply specifically the type of immunotherapy that is more likely to be effective in that disease and that patient.
Earlier this year, pharmaceutical companies teamed up for an ambitious clinical trial in the UK for a personalized cancer vaccine that targets specific mutations found in each person's tumor. At the same time, other companies announced their own version of a personalized cancer vaccine, reporting that the trials showed promise in pancreatic cancer, which has a notoriously poor survival rate. The vaccines work like this. Researchers take a little piece of the patient's tumor and a sample of healthy cells. They sequence both samples and play a game of spot the difference to hunt the genetic mutations that are exclusive to the tumor. We've already talked about this in episode 1 on DNA. Once they've got the mutations, they feed them to an AI algorithm, which then spits back a sort of police sketch of the tumor, ready to be handed over to the neighborhood watch group. This approach could help direct the immune system response right where it needs to go, like a GPS for our body's defense squad.
And there is a vaccine which is given now in teenagers that can protect more than 90% of the papilloma virus-induced cancer, such as cervix cancer. So basically, we can now use a personalized vaccination therapy in cancer.
Another approach would be to discover something that's a bit less tailored, but can catch different types of tumors. A kind of one-fits-all sweater that is comfortable enough for everyone, but stylish enough to work on a variety of occasions.
I mean, the final dream, of course, it would be to develop something that, in terms of immunotherapy, is capable to boost the activity of the immune system against molecules that are shared by different tumor types.
Of course, there's no guarantee that any of these strategies will work. Even if one method finds success with a particular type of cancer, that doesn't mean it will succeed with them all. But promise is in the air. And we've already got some pretty impressive success stories that suggest we're stumbling on the right path.
So I hope that in the next 10 years or so, efforts in all these directions, which significantly increase our tools actually to fight the cancers, and also we will learn how to apply them most effectively in each patient.
While digging into this episode, I couldn't wait to ask some of the world's top immune system experts about what we can really do to boost ours. And more specifically, this is what I wanted to ask. Are those immune boost pills I pop a few times a year actually doing anything? Or am I just fooling myself?
Vitamins, for example, are certainly important. For example, what is very well known, that deficiency of vitamin D, which is very often in people living in cold latitudes where you have to cover your skin because it's cold outside, and especially in the winter, very often a large proportion of the population has vitamin D deficiency. So, for example, supplementation of vitamin D is certainly important for the health of your body and also for the effectiveness of your immune response. But there are also some of these supplements that are not yet proven clinically that they are useful. And I think that we have to be very careful exactly what we are taking, and also consult eventually your family practitioner for advices regarding that, whereas a certain supplement is really important or not. And we have to be aware that unfortunately many of them have not been tested properly in large clinical trials to prove their effect.
There is really no strategy, no pills that allow us to keep our immune system healthy. For sure, a regular life with good intake of foods and so on, and an healthy lifestyle, I would say, is for sure something that helps our immune system. Many patients ask, what can I take in order to boost the efficacy of my immune system? And there is actually nothing that can be utilized, in spite of all the products that if you go in a pharmacy or in a drugstore, say, boost your immune system, take this one, these pills and so on. No way. A regular lifestyle is what is the most important part to keep it healthy.
It is also known that a very stressful life has a negative impact, and also, unfortunately, sometimes familial tragedies and so on have negative effects on your immune system. Sometimes we cannot avoid them, but if you can have, of course, a productive but at the same time balanced life is very useful.
So it's not just the microbiome. With a good life, we're getting to protect our immune system too. We're in charge of protecting our protectors. It's 2 a.m. as I finished writing this episode. I guess I'm not doing such a great service to my immune system. In this episode, we've only scratched the surface of proteins. For example, we mentioned those little molecules that hit the brakes on T cells, keeping them from going full blaze. Next, we'll dive into this fresh frontier of proteins and explore how they might help us pinpoint our risk of developing chronic diseases years before they even knock the door. The Science of You is a Cora Media podcast, supported by the European Partnership for Personalised Medicine, coordinated by Fondazione Toscana Life Sciences, and co-funded by the European Union's Horizon Europe Research and Innovation Programme. The podcast is written and hosted by me, Silvia Lazzaris, with editorial support from Simone Clemente and editorial curation by Graziano Nani. Sound and music supervision is by Luca Michele. Post-production and editing are done by Andrea Girelli. The sound engineer is Luca Possi. The project manager is Silvia Arcidiacono Mutti. The producer is Martina Conte, with post-production coordination by Matteo Scelsa.